Hello everyone, my name is Jack and I am a scientist here at Lhasa Limited. I am delighted to have the opportunity to speak with you about our degradation software Zeneth and how it can be used to solve real-world stability problems. To start proceedings, I'd just like to introduce Lhasa Limited. We are a not-for-profit organisation and we are driven by our purpose to enable informed decision-making on chemical safety. In line with this purpose, our objective is to create cutting-edge software technology which streamlines compound development and minimizes the need for animal testing. Our technology is designed by scientists for scientists, in collaboration with industry stakeholders and regulators. We develop knowledge-based, statistical and data-based software systems. The logos displayed here outline the suite of software solutions that we offer, and a full description of which each of these solutions does can be found on our website. Today I'll be covering an introduction to Zeneth, going through some background, the methodology of the software, and how we determine likelihood scores for degradation pathways. I'll then go through 4 use cases: degradant identification, structure elucidation of unknown degradants, how Zeneth can assist with nitrosamine risk assessments, and finally, how Zeneth can be used to support regulatory submissions. With that in mind, I will now present an introduction to our software solution, Zeneth. Firstly, I will address the need for Zeneth. There are specific guidelines outlined by the International Conference for Harmonization, or ICH as it is more commonly known. These aim to control different sources and types of impurities. The guidelines are Q3A, B, C, and D. There is also a new guideline for the control of extractables and leachables, with Step 4 predicted to be released in November this year. These guidelines help ensure that the drugs approved for market are safe and meet regulatory requirements. Zeneth can be used to help companies satisfy these guidelines due to its ability to assist with the various problems encountered when trying to understand the stability of a given drug. Zeneth is an in silico tool which can be used to predict what degradation is theoretically possible for a given chemical structure based on known degradation pathways. This will provide an overview of the theoretical degradation space for that compound, and therefore having this extra knowledge can support and aid decision making when undertaking forced degradation studies. Having a good overview of this potential space can ultimately aid understanding of the likely and actual spaces with respect to degradation products formed in final packaging and storage conditions. Zeneth is a knowledge-based system designed to predict theoretical degradation pathways. Its knowledge base can be thought of as a library filled with books on chemical degradation rules. Zeneth then uses this knowledge base to match these rules to a relevant fragment of a compound, after which a likelihood score is calculated for a potential degradant. The output of the system is a theoretical prediction of the chemical degradation possible for a given chemical structure. The knowledge base within Zeneth is now a mature 15 years old, and our last release contained 648 rules. We classify that knowledge into the following 6 categories. Oxidation and hydrolysis make up almost half the knowledge base. Condensations and additions a quarter, with the final quarter being split between photochemical reactions, isomerizations, and eliminations. I direct you all to the following 2 publications outlining validation studies which have been performed at different time points on Zeneth's knowledge base and demonstrate how the growth of the knowledge base, or our process of adding additional books to our library, has increased Zeneth's ability to accurately predict actual experimental degradants that have been observed in forced degradation studies. In addition to its knowledge base, Zeneth also contains an excipient database. Known contaminants and impurities are also listed against each excipient. We have a total of 368 excipients to date. Specifically, the database has nitrite listed as an impurity for 59 excipients. This information comes from the Vitek Nitrates database, which is a data sharing initiative hosted by Lhasa. This impurity information can help users when assessing the vitality of nitrate levels in a particular excipient for formulation, especially if nitrosamine formation is a risk factor. Having the ability to assess active pharmaceutical ingredients, or APIs, to excipient interactions is a key and valuable use case for many of our members. As a simple example, if we wanted to formulate cefuroxime with mannitol, as I've shown here, Zeneth would tell you that there's potential for an esterification reaction to happen. This may help drive decisions around whether it's acceptable to formulate cefuroxime with excipients containing primary aliphatic alcohols. I will now discuss the methodology behind the tool. Firstly, a user inputs a query structure into the software interface, i.e. your API of interest, as an example, this compound. You then also define the likelihood level you are interested in, so whether you want to know above or below a certain threshold. Meaning, if you only want Zeneth to tell you the degradants are likely to occur, or tell you everything that Zeneth knows about your input query structure. Finally, you also define the reaction conditions, such as whether water is present or by setting the pH of the system. The conditions available in Zeneth mirror those recommended in the ICH Q3B guidelines and are typical of those employed in forced degradation studies. Temperature and pH are numerical scales, and the other conditions, water, oxygen, metal, radical initiator, peroxide, and light, are either present or absent. All of these are easily set when running a degradation prediction. All of this information is collected and then used by the transformation engine, which cycles through the knowledge base, our library of books, to find matching transformations. Pattern matching in Zeneth works on a purely structural level, which is based on matching of functional groups or fragments of a compound. If a fragment of your input query structure matches the same fragment outlined in a pattern in the knowledge base, that is considered a match by the software. Each pattern match then undergoes a mathematical calculation to derive the likelihood score for that degradant being observed. And then finally, your product degradants are predicted by the software. All of these are displayed with their respective likelihood scores. The output from Zeneth is a tree of degradation pathways. This allows for the display of successive generations of degradants from the API or input structure. This is just a section of the results tree for the API bromazepam to serve as a representative example. I've mentioned that predicted degradants also get a likelihood score. We calculate the likelihood of a given degradant being observed using a scale from 0 to 1000, where 0 is impossible for that degradant to form and 1000 is certain that that degradant will be observed. We base the scores on variables such as reactivity, influence of reaction conditions, as well as using our built-in physicochemical calculators such as pKa and BDE, which I'll explain in more detail in the coming slides. To illustrate likelihood scores, the boxed figure on the left shows the likelihood scale we use in Zeneth 10. In older versions of the software, we used categories instead of scale. Whilst these categories were useful to qualitatively define levels of how likely we thought a given reaction would be, they did not allow for much granularity like a continuous scale does. So what we tend to do now is map the two, i.e. a score between 400 and 599 we would categorize as equivocal. A score between 800 and 999 we would categorize as very likely. For the example API shown, 4 hydrolysis pathways would be predicted by Zeneth, as well as a prediction of the structure for each degradant resulting from these hydrolysis pathways. An associated likelihood score of each of these pathways occurring is also calculated for the conditions ran. Because hydrolysis of an ester or a lactone is more likely to happen than the hydrolysis of an amide or a lactam in general, this has been reflected by the higher likelihood score predictions for the ester or lactone hydrolysis. When assigning a likelihood score, the software will consider some key factors. These include how readily the degradation reaction is expected to occur. This relates to the intrinsic chemical reactivity of the functional group in question. Secondly, the influence of reaction conditions. So if we are looking at an acid-catalyzed reaction, it will return a higher score in acidic conditions and a lower one in pH 7 or basic conditions. The same is true for temperature. For example, where a reaction is driven by heat, Zeneth will return a higher score if the prediction is run at 50 degrees when compared to a prediction ran at room temperature. The amount and quality of literature available for that given degradation pathway will also influence the accuracy of a given score. The relative position of the degradant within the pathway as a given score always takes into consideration the full pathway, and not just the individual steps. So if we are looking at a secondary degradant, its score will depend on its direct parent. Finally, and more recently, we are using physicochemical predictors to influence our likelihood scores. These physicochemical predictors calculate a specific property of a compound and use that value to influence a degradant's likelihood score. This allows the potential to predict more realistic and therefore more accurate likelihood scores. We are currently harnessing a pKa predictor which can be used to influence, for example, the likelihood of epimerization sites. In this example, there are two possible sites of epimerization. The epimerization at the carbon with a benzene substituent is predicted as more likely to happen when compared with the epimerization at the carbon bearing a methyl substituent. This is due to the different pKa's of the acidic hydrogens. In addition, we are also utilizing a carbon-hydrogen bond dissociation energy predictor to influence oxidative degradation pathways that proceed via hydrogen abstraction by a radical initiator. The BDE tells us the amount of energy required to break a given CH bond. In this example, there are two sites where benzylic oxidation can happen. One of the H's has a predicted BDE of 86.9 kcal per mole. and the other 89.3 kcal per mole. The lower BDE, i.e. requiring less energy to break the CH bond, results in a degradant of higher likelihood. In addition, we also have a chromophore predictor, which uses the size of a chromophore to influence the likelihood score for photochemical degradations. In this example, as the size of the chromophore increases, the likelihood of a photodehalogenation occurring also increases. The highest likelihood score correlates to the photodehalogenation of the naphthyl chromophore, followed by the anisole fragment, and finally the pyrrole possessing the lowest likelihood score. This directly correlates to chromophore size. Previously, all three of these chromophores would have been given the same likelihood score, since we didn't have a way to account for the size of the chromophore present. Having given you an introduction to the processes behind Zeneth, I will now move on to my next section, where I will detail some of the key Zeneth use cases and how our software can be effectively utilized. My first case study will outline how Zeneth can help elucidate the structures of degradation products observed in forced degradation studies to aid in the understanding of their mechanistic pathways. The scheme above outlines the experimentally observed degradation products seen for beclabuvir from forced degradation studies adhering to ICH guidelines. These degradants were characterized using NMR, high-resolution mass spectrometry, and HPLC techniques. From the scheme, we can see that degradation was observed in photochemical, oxidative, and acid-base hydrolysis conditions. Zeneth correctly predicted 7 out of 8 of these experimentally observed degradation products, those colored in green. All except D4, which is highlighted in purple, and is the hydrolysis of an N-acyl sulfamide group that is not currently predicted by Zeneth, but related chemistry is, and in the knowledge base, namely the hydrolysis of sulfamides and of N-acyl sulfonamides is. So expanding to cover N-acyl sulfamide groups will capture this chemistry in the knowledge base for future predictions. This is one way in which we identify and act upon knowledge gaps to build the coverage of our knowledge base in a relevant way. Furthermore, Zeneth doesn't just predict the structure of degradation products to help confirm analytical characterization. Mechanistic information, including intermediates, are also shown to aid in understanding. For example, in the formation of D2 and the diastereomers D3 and D5, Zeneth predicts the following mechanistic paths for their formations. These pathways are corroborated with literature evidence. In the formation of D2, this is proposed to go through an oxidative pathway via a 2+2 cycloaddition, generating intermediate 1. D3 and D5 are proposed by formation of a 2,3 epoxide, which then opens to generate intermediate 3. This is an alpha hydroxyaminium ion, which then undergoes a pinnacle type rearrangement, a 1,2 shift, to generate the dihydropyrrole 3-one product. All the literature information that was gathered to build the knowledge of that degradation mechanism pathway is displayed to the user as part of the prediction result. This enables the user to easily navigate to the primary literature if they require. For my second case study, I will show how Zeneth can aid in the structural elucidation of unknown degradation products, This is ideally suited to aid in the comprehension of the results of forced degradation studies. Stability indicating methods are used to resolve, detect, and identify the degradation products seen in forced degradation experiments, but they are complex and are costly to develop. Forced degradation studies on the compound BMS-644950 led to 8 degradation products. An HPLC method was developed to detect, resolve, and elucidate these products, but 2 of them could not be elucidated. D7 and D8 were identified as having masses of 500 and 466 daltons respectively. Zeneth was able to suggest theoretical possibilities for both masses, and if coupled with the additional experimental data, could provide a way to elucidate the structure of these unknowns. For the unknown degradation product D7 at 500 daltons, Zeneth predicts 6 possibilities. 5 N-oxides, each with a score of 700, and an epoxidation of the alkene, which was predicted as less likely than the other possibilities, at a score of 500. Zeneth can therefore provide a useful starting point for the user to map additional data and knowledge, which enables informed conclusions to be gathered from forced degradation trials on the actual structure of the unknown degradant. Secondly, for the unknown degradation product observed in acidic conditions, D8 at 466 daltons, Zeneth predicts 3 possibilities. 2 are predicted as very likely at a likelihood scores of 900. This high likelihood score is attributed to the fact that these pathways lead to the formation of conjugated degradant products, whereas the degradant with a likelihood score of 500 occurs from the dehydration of a secondary alcohol, which forms an unconjugated degradant product, which is reflected by the lower likelihood score. Theoretically predicted degradant products generated by Zeneth can be used to directly assist in structural elucidation studies. For the third use case, I will detail how Zeneth can assist with nitrosamine risk assessments. The risk of nitrosamine formation is, of course, something that has impacted many stages of the drug development process and many industries. To assist with this global issue, we have researched many theoretically possible nitrosamine-forming degradation pathways, and we have coverage of all the shown functional groups. For example, secondary amines which have appreciable nucleophilicity are often considered vulnerable amines, and will be predicted to nitrosate with a reasonably high likelihood, whereas some tertiary amines are much less reactive and require an initial dealkylation step before they can be nitrosated, so they are predicted at a lower likelihood level of formation. To assist with understanding all these theoretically possible pathways, we have implemented a specifically tailored nitrosamine risk assessment prediction in Zeneth. This is a pre-populated prediction set up containing conditions known to elicit or to propagate nitrosation reactions, such as acid or temperature, as well as excipient impurities which we have found from literature evidence that can lead to nitrosamine forming species. Including these impurities allows a user to assess all the possibilities of nitrosamine formation within Zeneth's knowledge base. The second step of this risk assessment is then an expert review of the results to correlate which impurities and conditions are relevant to your product stability study in order to assess the potential risk of nitrosamine formation. Zeneth is designed to predict the worst case scenario, so that nothing is missed and possibilities can be ruled out. So how is this assessment useful? What questions can it help answer? If we use Gramine as our example API, the nitrosamine risk assessment for this compound will predict that one of the theoretically possible nitrosamines that can form through degradation is N-nitrosyl dimethylamine, or NDMA. This is hardly surprising. There is plenty of published experimental evidence to show that NDMA has been found in many batches of Gramine. What Zeneth will provide, though, is all of the different routes and conditions by which NDMA can form. This, along with expert review of the results, can then help form part of a risk assessment for Gramine by helping to understand what mitigation strategies should be employed, as well as help direct which materials should be considered for formulation. I will now discuss 3 of the possible routes by which the formation of N-nitrosyl dimethylamine is possible and what mitigation strategies could be employed. Firstly, the nitrosamine risk assessment results for Gramine show that in acidic conditions it has the potential to first hydrolyze to an alcohol and dimethylamine, DMA, which in the presence of a source of nitrite at low pH will then nitrosate to NDMA. Zeneth predicts a high likelihood score for this pathway. Nitrite is present in many excipients, so one mitigation strategy would be to choose excipients with either no or negligible levels of nitrite in them. If, however, you're formulating Gramine in a liquid formulation, where pH is controlled during the whole process and a buffer is added to the formulation to ensure the pH will be stable, then this acid catalyzed pathway might be unrealistic and can therefore be ruled out. DMA can also form from Gramine via an oxidative N-dealkylation pathway at neutral pH in the presence of a source of nitrite and formaldehyde, whereby formaldehyde acts catalytically. So another mitigation strategy would be to choose excipients which do not have formaldehyde as well as nitrite as contaminants. Formaldehyde, like nitrite, is a well-known contaminant in many excipients. It should be noted that this is a less likely pathway of formation than the acid-catalyzed pathway. Finally, this oxidative N-dealkylation pathway can also occur in basic conditions at elevated temperatures and if there are any traces of an alkyl nitro compound, NDMA can result. Whilst this may seem like a lot of variables, it's easy to envision such conditions being present when considering storage conditions in warm climates. This knowledge can therefore be utilized when choosing packaging and storage conditions to mitigate this scenario from occurring. Before I detail our next use case, I would just like to highlight that this following work was done as part of Consult Lhasa, which is a subsidiary of Lhasa Limited. Consult Lhasa provides a consultancy service utilizing our science within Lhasa as well as our software solutions. The types of services we offer can be found at the consultlhasa.com website. This final case study demonstrates how Zeneth, coupled with expert review from our own scientists here at Lhasa, can solve questions posed by regulators to assist with submissions. So a company approached us with the following issue. They had identified this amide-containing impurity in their stability studies. This degradant was seen in long-term stability studies when heated in a basic environment. And so, upon submission, this customer reported that this impurity was a degradation product of their active pharmaceutical ingredient, API, but the regulator didn't agree and required a deficiency letter to prove that this impurity was not a synthetic impurity. The customer wanted to know if Zeneth could provide further evidence to establish that this impurity was in fact a degradation product of their active pharmaceutical ingredient. Using Zeneth, we ran predictions on their API and noted that this hydrolytic pathway was predicted. Here, the pyrimidine 4-one ring undergoes base-catalyzed hydrolysis under thermal conditions to generate carboxylic acid and amine products. Note that the conditions for this pathway mirror the conditions of the stability study the customer saw their impurity in. The mechanistic steps for this pathway are described in detail by Zeneth. Notably, there is an observed intermediate resulting from ring opening. Expert review noted that this observable intermediate could degrade via a different hydrolytic pathway to generate a 1,3-dicarbonyl compound and the amide impurity in question. Zeneth describes that this pathway can be catalyzed by base and has been seen in the degradation of mitomycins. But why then was this impurity not seen in the customer's degradation studies and only in their longer-term stability studies? It could be that the thermodynamic conditions of the stability study provided just enough activation energy to lead to the amide impurity forming from an accumulation of the observed intermediate via pathway 2, but not enough to lead it to form the acid-amine impurities in pathway 1. This was further corroborated by an assessment of the pH dependency and the likelihood scores which Zeneth predicts for both pathways. This information and the mechanistic proposal from the supporting literature evidence was then used in a deficiency letter to the regulator. The outcomes of this work were that Zeneth was able to derive a realistic mechanistic route for the stability conditions this impurity was seen in. The key outcome being that this argument was accepted by the regulator, and it allowed the impurity to be controlled as a degradant rather than a synthetic impurity. This then saved the customer from having to develop an additional analytical method during quality control, which would increase production costs, and this was all achieved using in silico assessment results. I'd like to end my talk by thanking you all for your attention. I hope I've provided some useful case studies to demonstrate how Zeneth can be utilized to solve real-world stability problems. I'll now be handing back for a live Q&A session. Thank you.